Hello, I am Dr. Gunamuni Rabha and he is my student, first year MBBS student Raja Agarwal. Today we are going to show you about the development of the heart and cardiac looping. So here they made, actually they are one group and they are about 10 members in their group, and they made this model about the cardiac looping and development of the heart. So they are going to explain about the development of the heart. So let's start now. Raja?
Yes, sir.
So name of these group members in their team. These are all about the models. So explain it. From how will you explain?
So our chief model is to explain the cardiac looping, but before going into it, I'll slightly like to explain about the formation of the heart. And to explain that, I'll be taking help of the models prepared by our respected professor, sir, Dr. Baneswar Maro, sir.
Okay.
So this is the model which shows the development of a blastocyst. So in a blastocyst, we know that in the upper region we have the amniotic cavity.
This is a cavity, no? This is like this is a spherical in shape.
Yes,
Okay.
spherical
There should be one cavity.
cavities.
Okay.
So in the upper region we have the amniotic cavity, and in the lower region we have the yolk sac.
Okay.
And between both the cavities we have the bilaminar germ discs. And bilaminar germ disc includes the ectoderm and the endoderm. We know this.
Okay. What this
So
one?
this is the bilaminar germ discs,
Okay.
which separates the two cavities, amniotic cavity upwards and the yolk sac downwards.
Okay.
So specifically speaking about the bilaminar germ discs.
In between this, that means this one is the bilaminar germ disc.
Yes, it lies between
Okay.
these two cavities and separates the two cavities, the yolk sac upwards, and so the amniotic cavity upwards and the yolk sac downwards. Now, this bilaminar germ discs, we see two foramen type structures here, one in the left side, one in the right side. Actually, this is not left right, this is cranial and caudal.
Okay.
So these two foramens are nothing but they are two regions where the ectoderm and the endoderm are tightly adherent. So this is the future oropharyngeal membrane and this is the future anal membrane.
Okay.
So we know that between the ectoderm and endoderm, as the embryo develops, we have the mesoderm.
Okay.
So when we uplift the ectoderm, we have the mesoderm.
Okay. That means middle one layer is the
Mesoderm.
mesoderm layer.
Yes, sir. Now this structure that we see here, this is the neural tube.
Okay.
Neural tube, and neural tube is overlaid by the notochord. This
Okay.
takes it to the notochord.
Okay.
Now we know that the intraembryonic mesoderm are divided into three layers. First of all, we have the paraxial mesoderm, then we have the intermediate mesoderm and the lateral plate mesoderm.
Paraxial, intermediate,
and the lateral
and
plate.
lateral plate.
So just lateral to the notochord lies the paraxial mesoderms.
Okay.
The circular structures.
These
The
are
circular structures
paraxial.
are nothing but the somites.
Okay.
These are the future somites. Then lateral to the somites of the paraxial mesoderms, we have the intermediate mesoderm. These white color structures.
Okay.
And lateral to that lies the lateral plate mesoderm. These red color structures.
Okay.
Now this lateral plate mesoderm is a horseshoe shaped structure which is joint in the cranial side and it is open in the anal side. Now this lateral plate mesoderm is divided into two. One is the somatic, one is the splanchnic,
Okay. Upper one is the somatic,
and lower one is
one
the
is the
splanchnic.
splanchnic.
Now these two are connected to each other. Now we see that this lateral plate mesoderm is fused in the cranial side but open in the caudal side or the anal side. Now this fusion, cranial to the cranial side, we know this is the cranial membrane. Cranial to the cranial membrane, this area is known as the cardiogenic area.
Okay.
And we need to focus mainly on this because we are studying about the development of heart tube.
Okay.
So cardiogenic area is an
That
area.
means we have to think about only in the cranial area of
Yes,
this.
sir. Because we are speaking about the cardiac development, so we'll focus on the cardiogenic area, and it is an area which lies cranial to the cranial membrane,
Okay.
and it is a fusion of the lateral plate mesoderm of either sides.
Okay.
So moving on, this is all about this model relating to the cardiac development.
Okay.
Now, further process lies in imagination of our minds. So we know that as the embryo develops, it undergoes two kinds of folding. One is the craniocaudal folding, and one is the lateral plate folding.
Okay.
So-
Two folding. One is the.
Craniocaudal folding and is the lateral plate folding.
Okay.
So now we have to imagine how this folding takes place. So we studied that the cardiogenic area lies cranial to the cranial membrane or
Yes.
the oropharyngeal membrane.
Okay.
So as suppose this was a blastocyst, now this folded laterally as well and as well craniocaudally as well. So this structure that we see here, this structure, it is the oropharyngeal membrane.
Okay.
And we saw here that the cardiogenic area lies cranial to the oropharyngeal membrane.
Yes.
But we now see that this area, this circular area, is the cardiogenic area. It is not lying cranially, but it lies almost back of the oropharyngeal membrane. Why does this takes place? It is because of the craniocaudal folding. Now, because the craniocaudal folding is taking place, this cardiogenic area is pushed towards the thorax cavity. So this area comes backwards. This is the cardiogenic area. Now, in the cardiogenic area, let's move towards the development of our heart, which is our actual topic. In the cardiogenic area, we have angioblast cells. This angioblast cells develops it into two structures. First of all, we have the endocardial tube and we have the pericardial cavity. Now, these two openings that we see here, this opening and this opening,
Yes.
these two are the endocardial tube. Two endocardial tubes on either side.
So from the beginning, that means how their orientation should be. This one.
Yes.
You explain like this way. So when is so, okay, that means endothelium. Yes. Okay.
So these two are the two endocardial tubes on either side of the developing embryo.
Okay.
Which is pushed towards the thoracic cavity because of the craniocaudal folding.
Okay.
And surrounding it is the pericardial cavity.
Yes.
Now, because of lateral folding, what will happen is these two endocardial tubes will fuse to form a common endocardial tube, and also the two pericardial cavities will fuse to form a common pericardial cavity. We can see here, after this structure, this is the lateral folding that takes place. It results in the formation of a common endocardial tube. This small oval shaped structure is the common endocardial tube surrounded by the common pericardial cavity. Now, this endocardial tube cannot lie in the pericardial cavity by its own. It needs to be supported by some structures. So this supporting structure is this band of fibers, which we call as dorsal mesocardium.
Okay.
So this was lateral folding, and I told that the craniocaudal folding results in the pushing back of the pericardial cavity towards the thoracic region. So we see here this orange structure is the pericardial cavity, which was lying cranial to the oropharyngeal membrane, but because of this folding, it is pushed towards the thoracic cavity.
Okay.
So moving on, we learned till now that we have a common endocardial tube which is absorbed inside the common pericardial cavity supported by dorsal mesocardium. So this structure now is our common endocardial tube.
Okay.
This common endocardial tube is segmented into four regions chiefly. So we can remember it by a mnemonic called BVAS, BVAS.
BVAS. Okay.
So what does B stand for? B stands for bulbous cordis, V stands for primary ventricle, A stands for primary atrium, and SV stands for sinus venosus.
Okay.
We can see through a schematic diagram as well. The common heart tube was this one, common endocardial tube. This will get divided into four regions basically: bulbous cordis, primary ventricles, primary atrium, and the sinus venosus. Okay. And truncus arteriosus is not a separate part, but is a part of bulbous cordis.
Yes.
We'll study about the development of each of these segments in further time. So moving on, we have this same structure: the bulbous cordis, primary ventricle, primary atrium, and the sinus venosus. And above this bulbous cordis, we have the truncus arteriosus, which is also a part of the bulbous cordis. Now, in the developing embryo, we obviously need the blood supply. So the blood supply enters through the sinus venosus, moves through all these segments, moves to truncus arteriosus, and moves through the dorsal aorta.
Okay.
Which is formed by the aortic sinus or the aortic sac.
Okay.
So now let's study about the individual developments of each of these segments. So as I already told, ES means aortic sinus or the aortic sac, which forms the dorsal aorta, through which the blood flows out of the heart tube.
Okay.
Then we have the truncus arteriosus. Truncus arteriosus forms the pulmonary trunk and the aortic arch. The bulbous cordis will form the right ventricle along with the outflow tracts.
Okay.
And the primitive ventricle will obviously name is implying. We saw that bulbous cordis forms the right ventricle. So what is left? The left ventricle. So primary ventricle will be forming the left ventricle. The primitive atrium will form both the right and left atrium. And lastly, sinus venosus is nothing but we studied that sinus venosus is a structure through which the blood flow enters the embryonic heart and leaves the aortic sac. So there must be obviously some veins that will be carrying blood to the primitive heart. So these veins are 3 in number on either side. We can remember this by a mnemonic called as VUC, from medial to lateral side.
PUC.
Yes, sir. So what does V stand for? V stands for vitelline veins, U stands for umbilical veins, and C stands for common cardinal veins.
Yes.
Now, in the future life, in the embryo, the left-sided sinus venosus that carries this VUC gets degenerated.
Okay.
All
Left
of these
side.
three gets degenerated, and it will be absorbed into the primitive atrium, which will be forming the future coronary sinus.
Okay.
It is not our main purpose right now, but just to explain, this the left horn does not persist in the embryonic heart, and it gets absorbed into the primitive atrium, forming the coronary sinus.
Yes.
And speaking of the VUC structures in the right-sided horn, it individually forms separate structures. Like the vitelline veins will form the inferior vena cava.
Yes.
The common cardinal vein will form the superior vena cava.
Yes.
And umbilical vein is a structure will be absorbed in the primary atrium.
Yes. Okay.
So this was about the BIVAS in the. Speaking of the period in which it actually takes place, it is the 4th week, or we can say around 22nd to 23rd day. So this common heart tube, the common endocardial tube, is formed in the 22nd day of the embryonic development.
Okay.
So now this was the development of endocardial tube and development of heart. Now our topic is development of cardiac looping. So before actually going to the topic, what is the purpose of cardiac looping? So does this structure, does this BIVAS model looks anywhere like the modern heart? No, because modern heart is somewhere different. We have the atrium towards the backside, the ventricle towards the forward, like that. But it is not like the modern heart. So because of the formation of modern heart, we have the cardiac looping, and also to establish outflow tracts. So what are the changes that takes place? We have the bulbous cordis and this primary ventricle. These two structures will start moving towards the right side, towards downwards and medially. So three
Right side,
directions.
downward,
Right, down and medially. And this primitive atrium will start moving towards the back side. Towards the back of the ventricle. We can see here, this is the 23rd day model. So we saw that the bulbous cordis and the primitive ventricle will start moving towards the right side. So bulbous cordis, ventricle moving towards the right side, towards downwards, and
bulbous cordis is directed to the right, but the ventricle, primitive ventricle, is directed towards the left.
Towards downwards and medially. The atrium, which was lying just immediately below the ventricle, is now moving towards the left side and towards the posterior region. So this, and followed by the 24th day. In 24th day, what will happen? This atrium will completely move back towards the ventricle with a slight area in the front, and it will start moving towards the upper region. It will start moving upward region.
Okay.
So bulbous cordis, primitive ventricle, primitive atrium, and the sinus venosus, which is still outflowing the blood into the primitive atrium.
Okay.
So this was cardiac looping. Going to the text, we can see that cardiac looping I forgot to mention a point that cardiac looping takes place because of a protein which is known as dynein protein.
Okay.
And cardiac looping, as we already saw, it takes place inside the pericardial cavity or the pericardium. Now this entire looping takes place in the pericardium, and the left horn of the sinus venosus merges and forms coronary sinus, which I already explained.
Okay, already
Draining
explained.
into the right atrium, because sinus venosus is attached directly with the primitive atrium. Now in cardiac looping, what we saw, the ventricular chamber moves front and below. We explained the ventricular chamber will be moving front and below, and the atrial chamber will move towards the behind and will move upwards as it progresses.
Okay.
So structurally, we can see this was the bulbous cordis. It will moving towards the downwards and the right side, and the primitive atrium moving towards the backward and upwards. So bulbous cordis occupies this structure, primitive ventricle will occupy this structure, and primary atrium will slightly move backwards, upwards, forming the structures behind the truncus arteriosus. This structure is what somewhat looks like our modern heart. So we studied that the purpose of cardiac looping is to develop the primitive heart into a modern day looking heart along with the outflow tracts. So this was our model, and we used Langman for serving our purpose, and this is the actual model of the Langman which we tried to copy.
Okay.
This shows the development of heart tube of 22nd day, 23rd day, followed by 24th day.
Yes. So nice explanation, Raja.
Thank you, sir.
Definitely, this was a good model, very nice model, and you explained very nicely. So I hope who are showing our videos, they will learn about the development of the heart, and if anything have to ask us, you can comment us in the comment box. And Raja definitely doing very well explained today, I hope. Thank you so much.
Thank you, sir.
Definitely.
Thank you, sir, for this opportunity, and we're forever grateful for you.
Okay. Thank you, all of you.